So basically today what we're going to be doing is we're going to be starting with Unit 3 revision or Unit 3 recap. So this is basically we're going to try to go from chapters 2 to 4 in today's session. I don't know how far we're going to get, honestly, but we'll try our best. So it's going to be more of a recap. So it's not going to be as in-depth as when we covered it in class, but yeah, it's just a quick recap. And if you've got any questions along the way, you can ask me now, or we will be having another Zoom session on Friday to kind of like for me to re-explain any Unit 3 topics you're confused about. So as we're going through these topics today, I just want you guys to write in your exercise book on a little sticky note, what are the topics you're still unsure about, so we can revise them or recover them again on Friday, okay? Alrighty, so having said that, so we started off this year looking at chapter 2, which was all about nervous system functioning. So obviously one of the first things we did last year in our transition week was that we actually looked at the nervous system and the structure of the nervous system. And we said that the nervous system is actually structured in a kind of hierarchy. So you've got like your central nervous system and your peripheral nervous system. And then your peripheral nervous system divides itself into somatic and autonomic. And then the autonomic then divides itself into sympathetic and parasympathetic. And so what you really need to know about the nervous system is basically how are the nervous systems different from each other? What are the differentiating factors? So for example, we know that the autonomic nervous system is involved with all our automatic functions, okay, like heartbeat, digestion, breathing, all the stuff that basically keep us alive. Whereas the central nervous system is responsible for kind of all those, integrating all the information because it's got the brain, because it's got the spinal cord, okay? The sympathetic nervous system, on the other hand, is responsible for the fight-flight response, which basically activates in times of stress or activates when we are faced with a threat. So the main thing you need to be able to do is be able to differentiate between the main functions of each of those kind of segments of the nervous system there. Okay, and when we looked at the nervous system, we kind of talked about this sense that all the functions that we can do as a result of having, our central nervous system and our peripheral nervous system. So things like movement, things like experiencing sensation. All of these things are possible because of the presence of two neurons called sensory neurons and motor neurons. Sensory neurons obviously help us to experience sensation, and motor neurons help with voluntary movement. And we talked about a memory chip that we use, which is SAME, and you've got the I in the middle as well. So sensory is also known as afferent, motor is also known as efferent, and then you've got the I in between the A and the M here, which I wrote on the board, which was basically the interneurons, okay? And the interneurons are in the middle there because they always connect the sensory and motor neurons, because sensory and motor neurons can never connect directly. They need some kind of a messenger or a mediator in the middle to connect them, and that mediator is the interneuron. Okay, so as long as you know the structure of this hierarchy, this diagram is a little bit different from the one in your book and the one in your notes. But yeah, it's generally as long as you know the structure of the hierarchy here and what the main functions are, you should be all good for this topic. Okay, so when we look into sympathetic nervous system, we kind of look into this in a little bit more depth because we do cover this a lot in stress. And so it's important for you to really understand what is the purpose of the sympathetic nervous system, okay? The sympathetic nervous system activates in times of stress. So remember we said S for sympathetic, S for activating in times of stress. It prepares the body for action, and it is at the heart of the fight-flight response. The fight-flight response cannot activate without the sympathetic nervous system. Okay, so the sympathetic nervous system basically either prepares your body physiologically or biologically to confront the threat or fight the threat, or to run away, which is to flee the scene or to flight. Okay, so this is known as the fight-flight response. Now, what is the parasympathetic nervous system then? The parasympathetic nervous system is basically a nervous system that activates in moments where stress or the threat is no longer present. So in other words, it activates or it calms the body after action has occurred. And the parasympathetic nervous system is what we call as a counter system. Okay, so it's a counter system because it actually helps to calm the body after the action has, after the threat has passed. The parasympathetic nervous system is not an isolated nervous system division. It can only operate when the sympathetic nervous system is there. So they're kind of like those two systems work in conjunction or in contrast to one another. Now, without the parasympathetic nervous system, we will probably always be stressed and on edge. And so without the parasympathetic nervous system, we are very likely to die. We're very likely to get like serious disease because of stress, okay? So the parasympathetic nervous system has to be there to bring your body back to a normal state. So remember, P for parasympathetic, P for the fact that it brings the body back to a peaceful state. So then what's this big diagram over here about? So this is basically talking about the physiological effects that are to do with the sympathetic nervous system and the parasympathetic nervous system division activating. So when your sympathetic nervous system division activates, we know that your heart rate accelerates or your heart rate increases. We know that your pupils get bigger in your eyes, so you can see more of the surroundings and see more of the hazards in the scene. Okay, so it's important for you to know at least 3 to 4 of these changes. Okay, and we call these biological or physiological changes or effects. And we're going to do a little practice question on this very soon. So we should be able to not only know what these are, but be able to explain how do they allow a person to deal with the stressor or to deal with the threat that is in front of them. Okay, so I'm going to give you guys about 3 to 4 minutes now to just kind of jot down some, you don't need to write like a proper answer here, just jot down some points you might talk about in a question like this. And remember, it says with reference to the physiological aspects. So here you're really talking, Here you're really talking about all those physiological symptoms or effects we talked about on the previous slide diagram there, like increased heart rate and stuff. So try to just jot down a response and try to link it to any scenario here. So I'll give you about 3 to 4 minutes to do this now. And remember, when you're doing this question, it's really important for you to link back to any scenario here. Otherwise, you will not be getting full marks for this question. So try to link back to the actual stressor that Annie is going through and link to the physiological effects she might be experiencing and how they might help her, okay? So the question says, how might it be helpful? Not how might it be disastrous or how might it be harmful? We're looking at how it's going to help her. Okay, so we've got another 1 minute on the clock. Okay, so this question is asking, although she's able to function in her everyday life, Annie is stressed about her driving test scheduled for today. With reference to the physiological aspects of Annie's stress response, give 2 reasons why this level of stress may be helpful when Annie takes her driving test. So remember, whenever you see the term physiological aspects or physiological effects of a stress response, we're talking about all those like symptoms. So increased heart rate, increased dilation of pupils, increased breathing rate, inhibition of digestion, all those little symptoms that we looked at on the previous slide in that kind of colorful diagram there are basically what we're talking about. So here, what you're supposed to do is you're supposed to choose 2 of those physiological effects or physiological aspects, and then explain how are they going to be helpful when Annie takes her driving test, okay? A lot of students in 2015, when they did this exam, they were able to name those effects, but then linking them to actually how they'd help Annie when she takes her driving test was the more difficult thing to do. So anyway, this is taken straight from the VCA examiner's report. You can see the average mark for this question was 1.8 out of 4. Most people, like you can see, got the marks for at least naming. So at least saying increased heart rate, increased pupil dilation of pupils, that would actually give them 2 marks by itself. So whenever you get a question like this and you're overwhelmed, don't stress, write something, because you can actually still get marks. It's not always a case of just getting 0. In fact, the majority of 0
would you just identify them and that's it?
To get 2 marks out of 4?
Yeah.
Yeah, if you just identified like Annie's pupils would dilate and Annie's heart rate would increase, that would give you 2 marks out of 4. Yeah, and the other 2 marks come from you linking to the scenario and you explaining how each one was helpful for her when she took her driving test. So obviously, VCA's examiner's report are sometimes annoying in the sense they don't give you proper, like an actual, like kind of proper answer in a paragraph, but as you can see, we can just form an answer from this. So as long as you mention 2 of the following. So you could say, for example, her pupils dilated, which means her pupils in her eyes got bigger, which allowed her to see more of the road and more of the hazards that might have been on the road. So she could kind of like swerve and kind of like dodge those hazards, or kind of make sure that she didn't bump into something. Okay, so being able to see more of the road is something that is going to be helpful for her when she takes her driving test, okay? It's going to prevent her from smashing or crashing into something and failing her test. Another thing you could talk about was you could talk about the increased direction of blood glucose to her muscles, which is going to give her increased energy. So she's not going to be behind the wheel like, blah, I feel so tired. She's going to be actually like active and ready to try her best in the driving test. You can even talk about the release of hormones like adrenaline, which makes her more alert and more focused. So even if she was a bit sleep deprived the previous night, when she gets into the driving test, she knows this is serious stuff. Adrenaline starts to pump and she's no longer drowsy or tired. She's ready to concentrate and ready to be as alert as possible. Okay, so you just need to basically name the physiological aspect, and then you need to link it to how it's going to help her on the driving test. Not how it's going to help her in general life, not how it's going to help in terms of a general definition, but in terms of how it's going to help her for the driving test, because that is our scenario kind of stimulus material there. Okay, so that's basically where your 4 marks would come from. Okay, so let's move on to the next topic. Okay, so our next topic is basically talking about conscious and unconscious responses and the spinal reflex. And so when we did this back in chapter 2, we kind of differentiated that a conscious response is any response that the person is voluntarily involving themselves in, which means that they are having the full control of doing that behavior, okay? An unconscious response is any response that is involuntary, it's automatic, and it's reflexive. And a lot of the time, these responses are actually mediated through the spinal cord, which means that they're not always in our conscious control. So when we prick ourselves on something, on like a little rose thorn or something, or we touch something hot, that withdrawal reflex, which is our ability to quickly move our hand away, even before we realize that, oh my God, I just touched a hot stove, that is an unconscious response, because that does not involve our conscious awareness. And remember, we talked about the reason that it has to be unconscious, the reason that it has to happen so quickly and so reflexively and so automatically, is to promote our survival, okay? Because the spinal reflex, which is an example of an unconscious response, actually has an evolutionary role in the sense that it does promote our survival as a species. Okay, so that's the main difference between the two. The conscious response usually always involves the brain, and for that reason, it does always involve our conscious control, and we decide whether or not we want to do it. So if I want to pick up a coffee cup to drink my coffee, I should be able to show that as a conscious response because I've decided I want to do that. That's goal-directed behavior. An unconscious response is a response that I don't decide. My brain doesn't have much of a role there, and the spinal cord actually has more of a role, even before that information can be processed by the brain. Okay, so the spinal reflex is basically mediated through the spinal cord before the brain can play a role. Okay, so that's basically our conscious and unconscious responses there. Oh God, why is the answer already on this? Should not be like that. Oh, anyway, never mind. Okay, so you were supposed to do this as a practice question, but I forgot to hide this answer. So now everyone can see it. It doesn't matter. I'll just explain the question. Question 6, a waiter brings Karen some coffee in a glass. Karen picks up the glass of coffee in her right hand. She decides that it's too hot to comfortably hold and puts it back down on the table after a few seconds. Using psychological terms, explain the role of sensory receptors and the brain in Karen's action of putting the glass of hot coffee back down on the table. I think we did this in class, if I remember correctly. The main reason why this question had such a low average score of 1.3 out of 4 back in 2010 is because a lot of students, as soon as they saw this and as soon as they saw the information about the coffee being too hot, they automatically assumed that it was spinal reflex. But there was something in the question that actually suggested that it wasn't spinal reflex. And some of you might have already picked up on this, and it's the fact, this sentence here that says she decides that it's too hot to comfortably hold. When you're deciding something or when you're making a decision, that always involves your brain, okay? That always involves you thinking about something and you consciously making a decision. So this cannot be a spinal reflex because it's representing a conscious response on Karen's part. She's decided it's too hot to hold in her hand. Okay, so this is just an explanation that just explains the conscious processing behind how Karen is going to put down that glass of coffee back on the table. Okay, so I think I talked about this when we did this question in class a few months back, but you don't have to talk about the area of the brain that's involved. So here I've talked about the primary somatosensory cortex processing the coffee as being hot, but actually, because we don't study lobes in depth anymore in Unit 3 and 4, you don't have to mention this information here, okay? It would be good for you to mention the primary motor cortex because that is where motor messages are produced. But in terms of the somatosensory cortex, you don't have to mention that part of the brain in your answer, okay? Anyway, this is on the school box. So if you want to go back and look at this answer in a little bit more depth, afterwards you can do that. Okay, I think we'll just keep moving on then to the next topic. So our next topic is the role of axons, dendrites, synapses, and neurotransmitters. So everything on this slide is basically everything you need to know definitions-wise for the neurons. Okay, this is what Vika want you to know in terms of the parts of a neuron. There are parts of a neuron. Sorry, what was that?
Miss, is this PowerPoint on Toolbox?
Yes, it is under the week 4 tile.
Okay.
Yeah. Okay, so these are basically the definitions you need to know for the Vika, like for the exam, okay, according to our study design. You don't need to know what a node of Ranvier is. You don't need to know what a Schwann cell is, even though you will often see these labeled on diagrams of neurons you find online or maybe on Edrolo. You don't need to know what the definitions of those two things there, okay? You do need to know the definition of what a neuron is. An axon is the part of the neuron that relays or sends the message. Dendrites obviously receive the message from the presynaptic neuron. Synapse is that gap between two neurons, which acts as a site of communication, and it's the place onto which the neurotransmitter is released. And neurotransmitters are just basically chemical substances that help to speed up the message that's being sent. And in doing so, it's actually enhancing the neurotransmission. Neurotransmission means the sending of the neural message. Okay, and we've learned about examples of neurotransmitters so far, like glutamate, dopamine, GABA, all of these are examples of neurotransmitters. Okay, so I think that's pretty straightforward. This is just basically talking about the definitions, but when you learn these definitions, you are actually satisfying that study design criteria, which is knowing about the role of the axons, dendrites, synapses, and neurotransmitters. So as long as you know what these are and you understand these definitions, you are also learning the role of each of these components of the neuron as well. Okay, also I've put a little diagram here because obviously, like I've told you guys before, and you had a question on this in your SAC as well, you do need to be able to label a diagram, okay? The basic stuff, like what a dendrite looks like, what an axon terminal looks like, where's the axon, all that kind of stuff. That's basic information. You should be able to label a diagram of a neuron, and it's stuff that, it's something you've been doing since last year anyway, so it should be easy peasy. Okay, so coming towards the end of chapter 2 now, we're looking at the lock and key process. So the lock and key process is kind of this idea that in order for neural information to be transmitted successfully, in other words, in order for a message to be sent across the synapse successfully, and in order for a neurotransmitter and a postsynaptic site to actually kind of successfully fuse themselves together and for the message to be sent, we need to be able to satisfy this process called the lock and key process. Now, the lock and key process suggests that every neurotransmitter has a specific distinct shape, and that before it actually binds onto the dendrite of a postsynaptic neuron or postsynaptic site, it needs to make sure that it'll be compatible to the shape of that postsynaptic site. So if you look at this little like basic, like, I don't know, paint kind of diagram you see here, this yellow thing here could be the neurotransmitter, okay? And this blue shape here could be the postsynaptic site, postsynaptic receptor site. So as you can see, the kind of like the shape of the neurotransmitter here is completely matching or fitting into or clicking perfectly into the shape of the postsynaptic receptor site. And because it's actually matching in and it's fitting in very perfectly there, we say that the shape of the neurotransmitter is complementary to the shape of the post-receptor site. And because of this, the message will be sent successfully across the neuron, okay? Now, if the shape doesn't fit perfectly, let's say that if the neurotransmitter shape kind of doesn't fit perfectly into the post-receptor site here, and there are bits of the neurotransmitter kind of hanging about, not really fitting in perfectly there, neurotransmission cannot occur. So that means the message will not be successfully sent across the synapse to the postsynaptic neuron. So we have to think of this as kind of being like a lock and key process, okay? If I take one of your house keys and I try to open up my house, it's not going to work because obviously the shape of the key is going to be different to the shape of the lock. In the same way that if you get a neurotransmitter and you try to fit it into a post-receptor site that doesn't have a complementary or matching shape, the two are not compatible at all, neurotransmission is not going to occur, okay? So in this case, the lock is the post-receptor site and the key is the neurotransmitter. So you have to be able to unpack that analogy, okay? The key is the neurotransmitter and the lock is the post-receptor site. If either one of those is not compatible with the other, neurotransmission will not be successful and will not occur. So it's important for the neurotransmitter shape to be matching to the post-receptor site so that they can bind successfully, they can fuse themselves successfully, and the message can be sent across the neuron. Okay, and obviously we know that if message is successfully sent, there can be two effects, inhibitory or excitatory, and we're going to learn about those in the next slide, I think. Yes, before that, I'm going to give you guys about two to 3 minutes now to just kind of jot down some points you might use for a question like this. I think we did give you a question like this in your SAC for the outcome one. But just jot down some of the key things you might mention in an answer like this. You don't have to write a whole answer, just some keywords. Just want to see how you guys are going with your keywords and all that stuff. So I'll put like about 3 minutes on the clock and you can start to jot down what you think some keywords or key ideas for this answer would be.
Miss,
Yeah.
you know the process, do we need to know it like in detail?
Just the main kind of bolded parts of that previous slide we looked at.
Yeah.
Yeah, as long as you know what's on that slide, like there's kind of 4 main ideas, which I'll show you in a minute when I show this answer. But there are 4 main ideas with the lock and key process. So as long as you know there's 4 ideas, that's all you really need to know.
So not like step by step, right?
No, it doesn't have to be step by step. So it can be in any order in your answer. It doesn't matter.
Okay, cool. Yep.
Okay, so I'm just going to kind of show you guys the 3 or 4 main things you kind of need to talk about when we talk about lock and key process. Okay, so obviously you don't need to do these in order. It can be in any order as long as the 3 components are there. The first thing you got to do is define the term. So the lock and key process suggests that chemical neurotransmission can occur if the shape of that neurotransmitter matches or is complementary to the shape of the receptor site. So what I talked about before, the shape of that yellow component there, which was the neurotransmitter, has to match the shape of that blue component in that little diagram there. So, okay, so both of them have to be matching or complementary in terms of their shape. Then you have to unpack the analogy. So you have to say which one is the lock and which one is the key. So the lock is the post-receptor site, aka the dendrites of the postsynaptic neuron, and the key is the neurotransmitter. That's basically what you have to say in a single sentence. That's you showing the VCAR assessor that you know what lock and key process is. You know which one is the lock and you know which one is the key, and you're able to unpack that analogy. The last thing you need to do then is then describe the effect. So if, let's say that the neurotransmitter and the post-receptor site do lock into each other, they do kind of click into each other, there's a perfect fit, there's a perfect match, what then happens? What's going to come out of that? So essentially what's going to happen is that the neurotransmitter is going to bind to the post-receptor site, and the neural message is going to be sent successfully across the synapse to the postsynaptic neuron. And this is going to have either of 2 effects. Number 1, it's going to have an excitatory effect, which means it's going to activate or stimulate the neural activity. Or number 2, it's going to have an inhibitory effect, which means that it's going to block or prevent or stop neural activity, okay, on the postsynaptic neuron. So these are the 3 things you need to do. Number 1, define lock and key process. And in defining it, you are going to unpack the analogy, say which one is the lock, which one is the key. Number 3, what comes out of the lock and key process? What's the effect of a key successfully opening a lock? Or what's the effect of a neurotransmitter successfully fusing or binding with a post-receptor site? And that is, it's going to basically have an excitatory or inhibitory effect on the postsynaptic neuron. Okay, so those are the 3 main things you need to include in your answer. And these are basically all mentioned on the previous slide as well. So as long as you know what's on the previous slide, you'll kind of be all good for an answer like this. Okay, so let's move on to the next topic, which is obviously kind of related to what we were just doing. So remember I said that whenever lock and key process happens successfully, so whenever the neurotransmitter is a complementary or matching shape to the post-receptor site and is able to bind successfully to the post-receptor site, what's going to actually happen is 2 effects can actually be seen. Number 1, the neurotransmitter will create an excitatory effect on the postsynaptic neuron, which means that it's going to excite and activate and stimulate the postsynaptic neuron, or it's going to have an inhibitory effect on the postsynaptic neuron. So it's going to depress, it's going to block, it's going to prevent activity in the postsynaptic neuron. And so we learned about this traffic light kind of memory tip to remember the 2 main types of neurotransmitters, or the 2 main examples of neurotransmitters, and which one is inhibitory and which one is excitatory. So remember, glutamate is the green traffic light, because glutamate, as we now know in a lot more depth, has everything to do with learning and memory. Without glutamate, learning and memory are not possible, okay? And you have to think about that when you're learning something or when you're committing information to memory, it's always going to be, you know, you're activating the neurons. You can't stop neural activity and expect to learn something. It's not going to work. So glutamate is always an excitatory neurotransmitter because it is always involved with the activation, stimulation, and the firing of neural activity, or the firing of neurons, okay? Because it's always involved with stuff like learning and memory. GABA, on the other hand, is our red traffic light, because GABA, which stands for gamma aminobutyric acid, prevents firing. It prevents activity within the postsynaptic neuron. And for that reason, GABA has more to do with kind of depressing neural activity, with preventing neural activity, with stopping neural activity. So as we're going to learn in chapter 13 when we do specific phobia, a lot of patients who have specific phobia, they tend to overthink. They've got all this neural activity. The minute they see a little piece of thread on the floor, they think it's a spider. And if they've got a phobia of spiders, their brain goes into overdrive. Their brain starts working like endlessly, like all these messages start getting sent. Oh my God, is that a spider? Oh my God, what am I going to do? And so people with specific phobia for that reason actually take GABA as a medication, okay? They take tablets, which turn into GABA in the brain, and they are able to then reduce that overthinking, reduce that kind of overdrive that the brain goes into by stopping neural activity at certain postsynaptic sites. Okay, so that's what GABA is about. GABA is the red traffic light, which stops or prevents neural activity, and for that reason is an inhibitory neurotransmitter. Glutamate is our green traffic light, which actually activates or stimulates neural activity, and for that reason is an excitatory neurotransmitter. Okay. Okay, and then we look at the inhibitory effect of GABA in Parkinson's disease patients. So obviously we learned about Parkinson's disease as being a neurodegenerative disease. Along with Alzheimer's disease, it's one of the two neurodegenerative diseases we look at in our Unit 3. So obviously Parkinson's disease affects the brain, and for that reason it affects our central nervous system. Specifically in the brain, it affects a structure that's in the midbrain called the substantia nigra. So the substantia nigra is a part of the midbrain, and it's involved in voluntary movement. So all the movements that we make on our own, by our own will, by our own decision, is because we all have a properly functioning and properly intact substantia nigra. Okay, now what happens in Parkinson's disease is that patients patients who have Parkinson's disease start to experience a degeneration or a breakdown of neurons in the substantia nigra. And for that reason, you can tell that obviously if neurons are starting to die and break down in the substantia nigra, there is going to be some kind of issues with the way these Parkinson's disease patients are going to be moving. Okay, so you'll notice they don't move in a normal way. The way they move looks very abnormal. It looks very like something's kind of wrong with them. And it's very obvious from looking at them from the outside. So one of the problems is that the neurons in the substantia nigra don't just die out, but the neurons in the substantia nigra also were the neurons that produce dopamine. Okay, so dopamine is an excitatory neurotransmitter, and it's basically involved with sending like motor messages. So not only are these normal everyday neurons dying out in the substantia nigra, they're the specific neurons to do with movement, okay? And these are excitatory kind of neurotransmitters that we're talking about here, like dopamine. And obviously this is going to make the movement issue even more obvious and even worse, okay? There's going to be a lot of deficits and issues with movement. To make matters even worse, it's not just dopamine that's dying out, but it's GABA that's coming in and further reducing or further destroying the already depleted or already reduced stores of dopamine in the Parkinson's disease patient's brain. So not only is dopamine dying out gradually, GABA is coming in and accelerating that destruction of dopamine producing neurons. And for that reason, the actual motor or movement related deficits don't just slowly happen, but they happen very quickly. And you'll notice that within a space of 5-6 years, a patient with Parkinson's disease can go from kind of walking kind of semi-normally to not being able to walk properly at all. Okay, so it's a very kind of progressive condition that they're experiencing. Okay, and we talked about having to know like some of the symptoms of Parkinson's disease as well. So we're looking at things like the motor symptoms and the non-motor symptoms. The motor symptoms are anything to do with movement. So we're talking about things like tremor. We're talking about things like taking small steps, which is also called shuffling gait. We're talking about things like not being able to have much control over your voluntary movements. So when they pick up a cup of coffee, they're shaking, you know, they're trembling. Non-motor symptoms are symptoms to do with stuff like memory. So they do experience memory deficits because, again, this is affecting their brain. They will experience a loss of sensation, so loss of smell, loss of taste, and they generally have issues as well with their short-term memory and concentration. So all of these are kind of non-motor symptoms because they're not related to movement, okay? They're related to other aspects of functioning. Okay, so that's basically looking at Parkinson's disease. And with that, we are basically done with chapter 2. So I'm not going to start with chapter 3 right now, but I will try to find a way to, I don't know, we'll see if we have to carry the recap into next week, then we'll just have to do that. But yeah, we'll continue with chapter 3 tomorrow. Yes.
Can too much glutamate cause the brain cells to die out?
Too much glutamate? No, because glutamate is an excitatory neurotransmitter.
Yeah, but okay, got it. All right.
Yeah, GABA is the inhibitory neurotransmitter. So too much GABA in a normal, healthy person's brain can cause issues with neural communication. Yeah, because unless you've got some kind of disease or condition that causes you to overthink or causes too much neural activity, like specific phobia, for example, having too much GABA in a normal, healthy patient's brain is a bad thing. It's a bad thing for neural communication. Yeah.
What about glutamate? Having it too much?
Having too much glutamate shouldn't be a bad thing, but like I said, it can be a bad thing because it can lead to kind of excessive neural activity. And what that means in general, easy kind of language, is it basically suggests that the person might be overthinking too much, or the person might be, you know, basically like ruminating. So basically thinking about the same thing over and over again. So too much glutamate isn't a good thing, but having too much glutamate isn't like super common as well.
Right.
Yeah.
Yep.
Okay, so with that, I'm just going to stop the recording here.